Okay, so let's go. Where did we finish? Where did we finish previous session? Can you tell me, please? Separation of the bases. Which page do you know? We want to talk about separation of plasmids, yes? Separation of plasmids. I think separation of plasmids. This was the picture I told I teach you. Yes? Okay. So, I told you in this picture that we can separate the plasmids. The plasmids can be extracted by two types. The first one is from the conformation. The second one was from the its size. Okay, so let's talk about separation on the basis of size. I told you or not? I told you this slide or not? I told you this slide? Yes, okay. I remember that. I didn't start this, yes? Not complete? Yes, I complete this structure. As I told you, the plasmids are smaller than the chromosome of the bacteria, okay? They are smaller. If we add some lysozyme with ETTA with 25% of sucrose, what will happen? We have partial digestion. We have partial digestion of the cell wall, and the cell membrane will stay intact. We call it a spheroplast. We call it a spheroplast. And after that, I told you we add Triton X100. Triton X100 is non-ionic detergent. Okay, non-ionic detergent. After that, what will happen if we centrifuge the cell extract? What will happen? The cell debris come in the bottom. We have large fragments of DNA, and we have some plasmids. We have some plasmids in the supernatons. Which part do we want? Which part do we want? Tell me. Which part we want the supernatons, yes? So we take the supernaton by the, for example, sampler and discard the plate. Okay, understand? Okay, look at this here. I told you that the the chromosome of bacteria are but when we do the process of extraction, the DNA of the bacteria can break somewhere. So maybe we have some nicks in the chromosome of the bacteria. What will happen if we have some nicks in the chromosome of the bacteria? What will happen? Do we have supercoils? No. Look at here. If we have some nicks in this open circular bacteria, we can't look at here. Look at here. We can't access the supercoil structure. Supercoil structure. But the plasmids, because they're so small, and they can't, they may not break. So the structure of supercoil will remain. So, so here, look at here. We want to extract the plasmid of a bacteria because there is structure conformation. Look at here. If we prepare the cell extract, what do we have? We have linear DNA. What is linear DNA? Can you tell me what is linear DNA here? Can you tell me? No found? Here. Look at and listen to me. In this step, we just prepare the cell extract. Okay, so we break the bacteria. What do we have? We have DNA of plasmids and we have DNA of the bacteria. Okay. So what do we have? We have the DNA of a bacteria and we have DNA of the plasmids. The DNA of the bacteria is so big. The DNA of the bacteria is so big. In the process of extraction, in the process of cell lysis, the DNA damage and will break. So what will happen? They become linear. So what is the linear DNA? The linear DNA are the bacterial chromosome, are the bacterial DNA. And what is this supercoil DNA? The supercoils are plasmids because the plasmids are so small and they remain intact and become supercoil. Okay? Okay, so all of you tell me, what is the linear DNA? What is the linear DNA? Tell me. Tell me, what is the linear DNA? Bacterial chromosome. You don't listen to me? Thank you, Nashwa. Thank you. Thank you so much. And can you tell me what is the supercoil bacteria, supercoil DNA here? The supercoil are, we can read it here. The supercoils are plasmids DNA. Yes? Do you understand up to now? So, at first, we add some NaOH. NaOH. What is NaOH? You all know NaOH. NaOH. What is NaOH? It is, we call it sood. I don't know what you call it in Arabic. We add some NaOH. قاعدة. Okay. Yes, we have some NaOH here. So we increase the pH. So we increase the pH to 12. So we increase the pH to 12. What will happen if we increase the pH? If we increase the pH, the linear DNA, look at here, the linear DNA breaks to one single strand DNA, look at here, breaks to single strand DNA. But the supercoil DNA, because this is supercoil and so intense, they didn't denaturate. So the supercoil DNA remain intact, but the linear DNA denaturate into single strand DNA. Okay, after that we add some acid. What will happen if we add acid here? If we add some acid here, the pH decrease. Yes, the pH become to 7. What will happen, Hames? Thank you. Lara. Thank you. The pH decrease, and what will happen? Look at here. The linear DNA denaturated and become together, tangledness. They become together and denaturated. Look at here. But what will happen to supercoil DNA of the plasmids? Nothing. They become intact because they're supercoils. What will happen if we centrifuge? After that, if we centrifuge, what will happen? The plate of linear DNA, because they tangled mess, they go in the bottom of the microtube. In the plate, we have linear DNA molecules, and in the supernatant, what do we have? We have supercoil plasmids. We have supercoil plasmids. Understand? So, which part do we want? We want supernatant. So we uptake the supernatant with the sampler and discard the plates. All of you understand? Thank you so much. So, look at here. Look at the another picture here. Look at here. This is cesium chloride density gradient centrifuge. Look at here. This is cesium chloride density gradient centrifuge. So we have one column which has cesium chloride inside, and we centrifuge, and because the density gradient parts of the cell extract, we have some parts of cell extract. Look at here. Look at here. All look at here. This is cesium chloride. This is cesium chloride density gradient. From this to this part, cesium chloride concentration increase. So this is the increasing cesium chloride. Can you see? Okay. After that, if we put our cell in the cesium chloride density gradient and centrifuge them, we have RNA here, we have proteins here, and we have DNA here. In the density of 1.7, we have DNA. In the bottom of the tube, we have RNA, and in the top of the tube, in the density of 1.55, we have what? We have proteins. So you can see in this picture, in the cesium chloride density gradient centrifuge, we can separate protein, DNA, and RNA. Okay. What will happen if we want the DNA? If we want the DNA, I can use, for example, I can take the DNA from the column, and use it. But now, what do we want to talk about? We want to separate the plasmid from the of the tree. Okay, listen carefully. What do we want? We want to separate DNA of the bacteria from plasmids. Okay. If we add some ethidium bromides, do you know ethidium bromides? All of you, do you know ethidium bromides? Ethidium bromides are intercalating agents. What does it mean? It means ethidium bromide goes through the DNA, the basis of the DNA, and after we light some UV to it, we can see the bond. Okay, so look at here. This is the normal DNA. This is the normal DNA double helix. If we add ethidium bromide to it, ethidium bromide is intercalating agent. It can go through the basis of the DNA, and after, if we have some UV lights, what will happen? They will have some lights. Look at it. Okay. So please tell me, if we add ethidium bromide, if we add ethidium bromide to the plasmid and to the chromosome of the bacteria, which density decrease more? Listen to me. Please answer my question. If I add ethidium bromide to a chromosome of a bacteria and to plasmid, what will happen? No, not pure. The density will change. Why? Fatima, why? Because the chromosome of the bacteria are open circular, but the chromosome of plasmids, look at here, I told you, the DNA of the plasmid is supercoiled. Yes? So this is supercoiled. We have less ethidium bromide in it. But if we had ethidium bromide here, we have so many ethidium bromides in it. Understand? Ethidium bromides can enter the plasmid of the bacteria so easily because this is not supercoiled. But the plasmid is supercoiled, so it can change the density. So look at here. All of you look at here. If we add ethidium bromide and going through ethidium bromide cesium chloride density gradient, what will happen? RNA was here, I told you. Protein was here, I told you. The linear DNA, the bacterial DNA, because it is open circular, change the density. And look at this. Go upper, but supercoiled DNA, because this is so dense and doesn't change the gradient, the density, stay here. Understand? Understand this? Okay, so what do we want? What do we want now? We want supercoiled DNA. We want plasmids. Yes? Where is the plasmids? Here. Look at here. Here is plasmid. This is supercoiled DNA. We can remove, look at the picture, we can remove the supercoil with a syringe. With the syringe, we can remove the DNA. Okay, understand? Understand up to now? Okay, so we remove the supercoiled DNA with a syringe, but what do we have in this liquid? We have ethidium bromide and we have cesium chloride. We should purify this liquid from ethidium bromide and we should purify this from chloride cesium chloride. So the first we should add N-butanol. What does the N-butanol do? The N-butanol allow layers to separate out. In the supernatant, we have ethidium bromide organic phase. In the bottom, we have DNA in aqueous phase. So, which part do we have? Which part do we want? We want this part, the bottom part, yes? So we discard the supernatant and we want this part. What do we have here? that we don't want. Here we have cesium chloride 2 that we don't want. We can use dialysis. You all hear about dialysis, yes? In the dialysis, we have some buffer. We want dialysis tube. And after that, the N-butanol remove ethidium bromide. Look at here, Marve. After this, if we remove a supercoiled DNA, we have two items that we don't want. One is ethidium bromide and one is cesium chloride. Okay, the first one we want to remove ethidium bromide. If we add N-butanols and centrifuge, we have two different parts. Ethidium bromide in the upper side, we discard that. In the bottom, we have DNA. Okay, but after this, we have cesium chloride 2. How can we remove that? By dialysis. Okay, we have tube of dialysis here. You can see it. And the cesium chloride diffuse into the buffer. And after the cesium chloride diffuse the buffer, we have pure DNA in the tube. We have pure DNA in the tube. Understand? Thank you so much. Anybody wants me to repeat more? Thank you so much. Thank you so much. So, I want you to help me to continue. So, listen please. After we use the dialysis, if after we use the dialysis, we have what? We have pure DNA. Okay. We have pure DNA. What should we do next? Would you please tell me? What should we do next? Anybody? Anybody tell me, please? What should we do next? We should concentrate the DNA. How we concentrated the DNA? In the previous sessions, I told you. How can we concentrate the DNA? By adding, no, Fatima. What did we add? To concentrate after the constant DDW. Before adding DDW, what did we add? Ethanol. Do you remember? The absolute cold ethanol. Remember? All of you. I think you don't prepare the lessons, you don't read. Why this? I told you for concentration of DNA, we add cold absolute DNA, absolute ethanol. Okay, what will happen if we add absolute DNA, absolute ethanol, cold absolute ethanol? The DNA will go down. We have plate of DNA. We should centrifuge in high speed, centrifuge in high speed, so the DNA will go out, will go down, and we have plate of DNA. And in supernatant we have ethanol. We will remove the ethanol and let it dry. I told you we should let it dry. Okay, after that we should dissolve the plate in what? Fatima, you told me we should dissolve the plate in? Thank you, DDW. And what? What did else I told you? In DDW and? Or in TE? Do you remember? No. In TE. Students, listen to me. Ethanol is for precipitation. Ethanol is for precipitation, and for concentration of the DNA, we add ethanol. Which type of the ethanol? Absolute 100% cold ethanol to precipitate the DNA. After that, we remove the ethanol, let it dry, and add DDW or TE to dissolve the DNA. Okay, understand? Thank you so much. Remember process, please. If you remember the steps, it's so easy because the steps are repeated in other extraction. You know, for example, in all the extract types, precipitation, use ethanol for precipitation. All type of W or TE dissolve the plate DNA. So it's the same in all. Understand? Understand up to now? I told you how. Did I told you how we can determine the concentration? Did I told you we can the concentration? Let me repeat my question. How we detect, we extract the DNA? Hello, can you hear me? Can you hear me? Okay. Okay, sorry. My voice was disconnected. My voice was disconnected. So, what was I talking about? Ah, I was talking about how can we determine the DNA that we extract. Listen to me. How can we determine the DNA that we extract? For example, you extract the DNA. Okay, you extract the DNA, but how do you understand that you did your good? How you can determine the DNA concentration or the um, qualification of the factory extraction? Understand my question? Let me repeat more. Listen to me. You extract the DNA up to now, okay? But how you can understand you did well, you did your job good. How can you understand that you extract the DNA? Maybe you have some problems in one of your steps and you don't gain any DNA. How can you be sure that you have some DNA? And how can you qualify that? How can you understand the qualification of the DNA? For example, you may have some contamination in the DNA. How can you understand you have some contamination in the DNA? Anybody tell me? No, Fatima. No. After centrifuge and add DW, we have DNA here. Okay, we want to understand the qualification of DNA after that. Okay, do you know the process of spectrophotometry? Did you know about the spectrophotometry? Yes. What do you do in spectrophotometry process? In you can't hear my voice? I told you. I told you. A spectrophotometry process. Um, do you have my voice better? Do you have my voice better? No? Okay, let me refresh. Let me refresh, please. Hello? Hello, can you hear me? I'm sorry, there's so many problems. My voice couldn't connect. I refreshed so many times. Refreshed so many times to access my voice. Okay, you have my voice clearly? Okay, thanks God. I told you after we have DNA, we should determine if we have DNA or not, or we can determine the qualification of the DNA. How? By the spectrophotometry process. By the spectrophotometry process, how we have two different instruments. For example, nanodrop, or picodrop instrument. Sorry, I read it wrong. Oh, nanodrop and picodrop. So this is the two different instruments for detecting the DNA or determination of the DNA qualification. If we detect the OD in 260 nanometers, we can detect DNA. So in the wavelength of 260 nanometers, we can detect and determine the DNA amount. For example, we can determine that we have 1000 nanogram per microliter, nanogram per microliter DNA. So we can understand the amount of the DNA. How? By the use of the spectrophotometry instruments, for example, nanodrop or picodrop. In the wavelength of 260, we can understand the concentration of DNA. We can understand the amount of the DNA. For example, 100 nanogram per microliter, we have DNA. And in the wavelength of 260 to 280, we can understand the qualification of the DNA. How? We can understand if we have any contamination in the DNA or not. For example, do we have any contamination of proteins or not? So in the 260 to 280, we can understand the contamination. How? It should be about 1.8 to about 1. Do you have my voice? I don't know what happened to Dawn. I was disconnected and I couldn't enter the class. It really gained all my energy. I don't know. I am so angry. Okay. So, what was I talking about? I was talking about the 260 to 280. It should be about 1.822. Okay. The best number, it should be 1.9 to best qualification of the DNA. So in the wavelength of 260, we can understand the amount of the DNA. We can understand the concentration of DNA in the percent of 260 to 280. If the number is 1.822, it's the best qualification, and the best number is 1.9. Okay, if you don't have any question, let's go to the next step. The next step, if you can see, before the purification of the plasmid, listen to us a minute and help us. Yes, of course. What's your problem, Fatima? Help you summarize. No, it's not difficult, Fatima. All the students in Iran, which I teach them so many years, they understand and memorize the book so well. So, as I know you're the very good students, all of you are good students, you can understand the book easily. You should practice. You should study repeatedly, not just once. Okay, not just once. You should repeat it. Take a note. Please. Study the book and take a note for yourself. And after that, the second step, study your notes. Okay, and look at the pictures. The pictures are very helpful to understand the process. The pictures are very helpful. Okay, you can understand from the pictures. So, take a note, repeat reading your note. After that, you should memorize it with the picture. No. Because you didn't study, Marve. Because you didn't study, you think like that. If you take a note for yourself, no, this is not in the lab. This is not the lab. You should learn it in the theory, after that, if you want to do it, you go to the lab. Okay, this is not the lab work. I summarize the substance. I summarize the steps. You know, I'm teaching the whole book for the uranium students. But where do we now? We are just in the chapter 3. Look at me, students. As you know, as I told you, this is not necessary to understand it in lab. I didn't do all this step in the lab too. I didn't do it too. So, listen to me, please. Listen to me, just. I didn't do all this step in the lab because we do the professional and very specific ways. For example, if I do something in genetics, I didn't do all about the plasmids. So, but I should know about it. I should know the process. Oh, I write it to us for no, I couldn't. Sorry, I don't have the time to write it to you. So you have book and it's clearly explain it to you. And the book is so simple, and I think I repeat all these pictures 2 or 3 times. If you listen in the class, you will understand. If you listen in the class, you will understand. So, yes, I explain all the book for you up to now. Okay? I explain all the book up to now. You should, listen to me, you should study the book, take a note, and repeatedly read your notes by the help of the pictures. Okay? By the help of the picture. It's so easy. I promise, it's so easy. Just, it needs you to repeatedly study the book. Repeatedly, not just once. You don't study, Fatima. I'm sure. Why? Because in the previous, in the last minutes, I asked you some questions and you didn't answer well. Why? Because you didn't understand. You didn't study well. If you take a note and understand the lesson, it's so easy. So let's have a quiz. I think I should I should take a quiz. Yes? Next week, all of you study well from the beginning up to now. And next week, we have a quiz. I want you to understand well. Okay? If you don't understand me, Fatima, please ask me to repeat. I told you. I'm here to help you to understand better. Okay, if any times you don't understand, you can ask me or refer to book. Okay, no problem. Don't worry. Next week we have the quiz. Okay, all of you study for the next week. We have quiz up to now. I'll repeat it again. Please take a note. Study, take a note by the help of the pictures. By the help of the pictures, take a note. Memorize it with the picture. Okay. The next step. Listen to me, please. Listen to me, please. Here, what do we have here? What do we have here? This is the bacteria. This is the chromosome of the bacteria. And what is here? This is the plasmids. You can see this is the plasmids. If we want to extract the plasmids, we want more, so many plasmids, yes? If we increase the plasmids in the number, if we increase the plasmids number, look at here, we can extract them better. Look at here. For example, in the normal cells, we have about 20 plasmids. Look at here. We have 20 plasmids here. If we want to extract the plasmids, it's better to increase the plasmids. Look here in the picture, we increase the plasmids more than 1,000. More than 1,000. How can we increase the plasmids? Listen to me. How can we increase the plasmids? If we add some chloramphenicol. What is chloramphenicol? Help me, please. What is chloramphenicol? What is chloramphenicol? Antibiotics. Thank you, Marve. Thank you, Natek. This is the kind of antibiotic. What does antibiotic do? The antibiotics doesn't let the bacteria to amplify. Yes, inhibit the bacterial amplification. So, if we add some chloramphenicol, the chromosome of bacteria doesn't amplify, but the plasmids amplify too. So, if we add some chloramphenicol, what will happen? If we add some chloramphenicol, it doesn't let the proteins of the bacteria to synthesize. It doesn't let the protein of the bacteria to synthesize, so the amplification of the bacteria inhibits, so only plasmids can amplify. So we have so many plasmids in the cells. Understand? Thank you. I explain you in so simple words. So I hope all of you understand. You don't want to come to Iran for your thesis? No? It's so good if you come here and we have the class in the university, so you can understand better. So much different in the presence class in the university and the online classes. But for the thesis, I think you can come Iran and have the thesis, because I think in Iraq the facilities are we have shortage of facilities in Iran, in Iraq, I think. Okay. If you don't have any questions, let's talk about extracting the phage, extracting the phage suspension. Okay, let me explain. Look at here. If we want to extract the phage, look at here. If we want to extract the phage, so phage is so lighter than the bacteria. Yes, phages are so lighter in the weight. In measuring the purity of DNA strand, ratios 1:8. If less indicate contamination. Yes, thank you. I told you that. 260 to 280. Yes, Fatima. I told you in the previous slide. No, I don't know the first term, the next term. I think it should be online for you. It should be online for you. Maybe with RNA, we have contamination with RNA. Fatima. We hope this. Okay. If you want to be online, it's so easy for you because you're in your country. But if you want to understand better, if you come to the class, you can understand better. You know, this is two-way. We will be so happy to see you here, Mr. Ahmad. I know it's difficult. I know it's difficult. It's not my decision. The university principal's decision is to they inform you to come to Iran or not. But in present time, it's online. Okay, let's talk about extraction of the phage. Look at here. I told you what? I told you the phage are so lighter and the bacteria are so heavier. Yes? If we centrifuge the suspension of bacteria and phage, what will happen? Bacteria are heavier. So, what will happen to the bacteria? They're so heavy. Look at here. Plates of bacteria, and in the upper side, what do we have? We have phage suspension. Suspension. So, listen to me. If we have a culture with bacteria and phage, if we centrifuge, the bacteria are heavier. Yes, the bacteria are heavier. So, if we centrifuge, the bacteria, because they are heavier, go down. And what is in the upper side? We have what? We have phages. Okay, understand? Thank you. So, listen to me. Listen to me. We want to extract the phage. Okay? We want to extract the phage. As I told you before, we have lysogenic phages and we have lytic. So we have two ways of the phages lives. We have prophage, which is in the cycle of lysogeny, and we have lytic here, which lyses the bacteria. Look here, this is prophage. I told you it doesn't lyses the bacteria. It doesn't lyses the bacteria. And when the bacteria amplifies, they go to the DNA of the bacteria, but when the lytic phase is activated, they amplify so many DNA and synthesize particles of virus phages, and they lyse the bacteria and go out. Do you know this? You all know this lytic and lysogenic process? You all know? Thank you. Okay, listen to me and answer my question. Listen to me and answer my question. If we want to extract the phage, which phase do we want? We want our phage to be in lysogenic phase or in lytic phase. So, listen to me. Listen to me. Why, Fatima? Ali, I don't understand what you write. Lytic and lysogeny. Lytic or lysogeny? What did you write? Lytic? Lytic phase. Thank you, Ali. Thank you, Hale. Why lytic phase? Because we want the phage. We want the phage particles. If it's going inside the chromosome of bacteria, we can't access to the phage. So we can't extract the phage. So if we want the phage particles, if we want this, and if we want to extract the phage, which cycle do we want? Lytic. So how can we lead all the bacteria to the lytic phase? Listen to me, all of you listen to me. We can use a phage with a mutation. What is the mutation? The mutation, look at here. We have the mutation in the phage, which induced by the temperature. Look at here. We have one gene which is C1 gene. Okay. We have one gene. The gene names is C1 gene. Okay. The C1 gene is the gene which is responsible to maintain the phage in the lysogenic process. So, the C1 gene is responsible for remaining or for maintaining the phage in the status. If we have the mutation in gene C1, we call it C1 temperature sensitive. Temperature sensitive. What does it mean? It means this gene has the mutation which is sensitive to the temperature. So, the C1 gene is functional in the temperature of 300 degrees, at which if we increase the temperature to 42 degrees, this gene cannot work properly, so the lysogenic process cannot be maintained, so it will go through lytic phase. So let me repeat again. If we want to extract the phage, which cycle do we want? Repeat after me. What cycle do we want? We want lytic phase. Yes, yes, we want lytic phase. So we can use a mutation. Listen to me. We have one gene. This gene name is C1. Okay, what does C1 do? C1 is the gene which helps the phage to remain in the lysogenic process, lysogenic phase. So, in the normal, this gene maintains the phage into the lysogenic process. After we put some mutation in it, we make it to C1TS. It means it is temperature sensitive. If this is 300 degrees, it is doing properly and it's going to lysogenic process. But if we increase the temperature and we have 42 degrees, it doesn't, this gene, it doesn't work properly, so it go to lytic process. Understand? Fatima, listen to me. Listen to me. If we want to clone a gene, okay, at first we may not want the phage to go through lytic. At first we just want to clone the genes. So we can use the phage which has the mutation in gene C1. What is the mutation? The mutation is temperature sensitive, which this gene works properly in the temperature of 300, 1300, but if we increase the temperature and increase the temperature to 42 degrees, it doesn't work properly and it goes to a lytic process. So listen here. At first, this is the phage which is C1 temperature sensitive. I told you that C1 gene is responsible for lysogenic process. If we bring it to condition of 300 degrees, it goes to lysogeny. But if we increase the temperature, because this is sensitive to temperature, it doesn't work. So if it doesn't work, what will happen? It will go to lytic phase. Understand? Thank you so much. Okay, thank you. We have makeup class. I think it's 1:30 in the afternoon. So please be aware of the class. And thank you so much. Thank you so much. See you then. See you then.